Hi everyone, my name is Julie and I'm going to go over an overview of SARS CoV-2. In this overview, I'll be going over 4 topics: the source of SARS CoV-2, the molecular features of SARS CoV-2, the mechanism of SARS CoV-2 infection, and an array of COVID-19 symptoms. First, let's talk about how scientists were able to determine how SARS CoV-2 spread to humans. To do this, they first collected samples of pulmonary fluids and isolated viral cultures from 9 hospitalized patients who were diagnosed with an unknown viral pneumonia. These samples were analyzed to determine the genome sequence of said unknown virus and then compared to known viral genomes. In their phylogenetic analysis, Liu and other researchers found that all of the samples were closely related to the BAT-SL-CoV ZC45 beta coronavirus due to genomic similarities. Using the genome of the specific strain as reference, the genomic data isolated from those hospital patients were further analyzed. The comparison of BAT-SL-CoV ZC45 beta coronavirus and the patient viral sample genomic data is shown here. As you can see, each individual sample has very few deviations, at most 2 for any single strain, from the consensus BAT beta coronavirus genome, suggesting that it was likely that SARS-CoV-2 originated from a bat reservoir. This is figure 1A of the review that mentioned this previous study, summarized data in this phylogenetic tree. This tree shows SARS-CoV-2 is more closely related to the BAT-SL-CoV ZC45 and BAT-SL-CoV ZXC21 compared to the SARS-CoV that was responsible for the early 2000 SARS outbreak. This means that it was from the bat reservoir and not a mutation in SARS-CoV between humans that we got SARS-CoV-2. Now on to the molecular features of SARS-CoV-2. Genomic studies showed that the SARS-CoV-2 genome was split into several parts. The first 2/3 of the genome consisted of open reading frames 1A and 1B, which code for polyprotein 1A and 1B, respectively. These polyproteins are further processed to create non-structural proteins 1 through 16 of the virus. And the last 1/3 of the genome mainly codes for the 4 main structural proteins, aside from some accessory proteins. As I just noted, the first 2/3 of the SARS-CoV-2 genome code for what will eventually become 16 non-structural proteins that have various functions for the virus, as summarized here from the HAPMAL 2020 study. Although NSPs don't contribute to the structure of the virus, they have many functions, from facilitating viral replication, as shown with NSPs 12, making that RNA-dependent RNA polymerase, and NSP 14, for viral genome proofreading. And then there's also modulation of the host immune response as seen with NSPs 1 and 3, inhibiting host interferon signaling. The last 1/3 of the SARS-CoV-2 genome code for 4 key structural proteins, the S, E, N, and M proteins. The proteins are sequenced in this order on the SARS-CoV-2 genome, and their functions are briefly described here. The S protein plays an important role in viral host binding, as it interacts with host cell ACE2 receptors to facilitate entry into that cell. The S protein complexes with other S proteins to form a homotrimer that is compatible with ACE2. The E protein is found in the envelope of a viral particle and has a role in the production and maturation of the virus once replication begins within the host cells. The N protein is responsible for binding and organizing viral genetic material, and the M protein also adds to the structure of the viral particle membrane, especially functioning to hold everything together and determine the overall shape of the virus after it's assembled. Next, I'll be going over the mechanism of the SARS-CoV-2 infection. This schematic here is a part of the graphical abstract from the review by Lee and other researchers in 2020. To pull the snippet apart, infection starts with viral binding to host ACE2 receptors. This was found in a study published in The Lancet by Liu and other researchers after analyzing the genomic sequence of SARS-CoV-2 and observing how the SARS-CoV-2 binding domain had a very similar sequence to that of SARS-CoV from the early 2000s outbreak. This suggests that the binding receptor for SARS-CoV-2 was very similar to SARS-CoV. After viral recognition of a host cell through ACE2 binding, the S protein gets proteolytically processed to facilitate membrane fusion, releasing viral positive sense RNA into the host cell. Once in the host cell, the viral mRNA can be translated via host cell machinery, which will produce viral replication proteins to produce more viral particles. These viral particles are packed together between the endoplasmic reticulum and Golgi into vesicles that once again fuse to the host cell membrane, but this time releasing the viral particles out of the cell rather than bringing viral genetic material into the cell. In this final part of my summary, I'll go over some common clinical symptoms associated with the COVID-19 infection. Although many who are infected have a wide range of disease severity, the most common symptoms of an infection are fever and dry cough. Of the patients who were symptomatic and admitted into the hospital during a hospital study, about half of the patients who had fever already when admitted, and the percentage of patients with fever increased to almost 90% during the hospital stay. A majority of patients who were symptomatic and admitted to the hospital also presented with a dry cough, meaning that there was no production of mucus or sputum from this cough. Some other fairly common symptoms of COVID-19 infection in general also include myalgia, known as general muscle pain, dyspnea or difficulty breathing, and interestingly, anorexia. COVID-19 infection was generally known to result in a loss of taste and smell, which greatly reduced the pleasures and cues that can motivate a person to eat. Along with this, the discomfort of difficulty breathing and general pain may have further reduced the will to eat in many patients. Several studies covered SARS-CoV-2 through the year of 2020. It appears that much information was first obtained from genomic analysis, which really highlights the importance of genome sequencing in eliciting the characteristics of new viral species and pointing future studies in the right direction to learn more about viral mechanisms and potential targets for immunization of treatment. So that concludes my summary for SARS-CoV-2. Thanks so much for listening. And as application, this template was from these creators here. Thank you.